Hello friend, here is a vectorless DNA transmission. Vectorless DNA transmission means there is no external agent required to transfer the DNA into the host cell. So, in previous class we used a vector like plasmid, cosmid, vac, yak, phasmid, bacteriophage, etc. So, that carries the small as well as large segment of the DNA. So, in previous classes, vector involved to carry the DNA from one organism to another organism, and before the vectors there are various type of enzymes used to withdraw the DNA or gene or chromosome. So, first the enzymes are which break the outer boundary of the cell, that is cellulase, chitinase, lysozyme, etc. These are the various type of enzymes first which break the outer boundary. Then enzyme which act as a nuclear level which dissolve and digest the nucleus or inside the nucleus, that is chromosome, gene, DNA. So, that enzyme is called nuclease enzyme. So, a nuclease enzyme might be exonuclease which act on the terminal part of the DNA from 5' to 3' terminal. So, another is endonuclease which act on the inner side except 5' and 3' terminal, and one of the specific enzyme called restriction endonuclease which acts on the palindromic side. So, palindromic side is the side where the forward nitrogenous bases and backward nitrogenous bases are similar or shaped. So, that the point is called restriction endonuclease. So, there are the various site of restriction endonuclease where act. So, it is EcoRI, Hin2, Hin3, etc. So, where the action of the restriction enzyme acts. So, it may be or might be 4 to 6 nucleotide that produce two end. First is an unequal strand, that is called sticky end, and another one is similar nucleotide, both the nucleotide strand is called blunt end. So, after the formation of these action of these enzymes, another enzymes also that is called synthetase enzymes like DNA polymerase, RNA polymerase, and reverse transcriptase, and as well as some nuclease enzyme which act on the specific point, that is phosphate end, that called alkaline phosphate. So, after the action of this enzyme we withdraw the DNA. So, there is the various carrier we use to carry the DNA from one organism to another organism. Now we are using the vectorless DNA transmission where the direct DNA transfer. So, there is the process to transfer the DNA from one organism to another organism with the use of some tools. This use first is micro injection. It is just a injection of the DNA into the plant cell or animal cells called micro, where we use the micro injection like micropipette which enter the nucleus or specific DNA into the plant or animals. Especially in the egg cell, in the case of test tube baby preparation formation, test tube baby, so transfer of the male gametes into the female cytoplasm. So, this is the micro injection. Another one is electroporation, and in electroporation, so cells we form a pore over the local area of cell membrane or cell wall that form a pore, and then we enter the genetic material into the cytoplasm or nucleus into the shape. Third one is gene gun or biolistics. So, gene gun is a type of impulse technique where the DNA is coated with the tungsten and high velocity impulse applied there and makes the or with high velocity and it come into the cell cytoplasm. So, this is the gene again, and here the direct DNA transfer, where the direct transfer of DNA or mainly vaccines are introduced into the cells. So, this is the process of vectorless transmission, vectorless transmission. So, this is the tools, and when the tools enter into the competent host. So, how the tools we are using and in our transfer of DNA from one organism to next another organism, this is called, now it is technology. So, first the technology applied, here is the cells or this is the cytoplasm and nucleoplasm. So, the nucleus present in DNA present in nucleus or genes present in nucleus. So, in technology, in biotechnology, we are just withdraw the same sequence of the RNA instead of DNA, because DNA present inside the nucleus. First we break the nucleus, it is quite enough, but it is easiest to withdraw the RNA from the cytoplasm and withdraw RNA which convert the DNA through the reverse transcriptase process, and formation of the desired gene takes place by this technique, and desired genes which carry over through the carrier DNA, that is plasmid, and plasmid which combined with the desired DNA to form the chimeric DNA, chimera and or recombinant DNA. Then the chimera which introduced into the another living organism through the process of transformation, and it is called competent host, and competent host which multiply after introduction, multiply to through the multiplication or binary fission. In case of bacteria, it is multiplied 2, 4, 6, 8, 16, 22, so on. So, it also the genetic recombinant DNA also multiply, and recombinant DNA which contain the desired gene which produce the protein at mark level. So, we have produced the DNA product or gene product or protein at huge amount. So, this is the technology. So, technology used in recombinant DNA is first the formation of somatostatin. It is formed through the genetic recombination process with the help of E. coli, and somatostatin is just a growth hormone which is supplied through, which is produced through hypothalamic pituitary control, but this process little amount of the somatostatin we get, but the technology process, recombinant technology, biotechnology, genetic engineering process, we produce huge amount of somatostatin. So, this is the biotechnology. At the second was interferon, one of the protein which help with the defense process. So, interferon act as an antiviral and anti-cancer. It is a very potent proteinous structure we formed through the DNA genetic recombinance process or genetic engineering process with the help of bacteria through this technology. And the another factor 8 which is anti-clotting factor, it is factor 8 means clotting factors. So, clotting factors we produce through DNA recombinant technology. And finally, insulin is a major proteinous structure help in the maintain the glucose level. If synthesis or level increase in blood, it goes to hyperglycemia and person shows the diabetic. So, this is the insulin which is formed through the genetic recombination technology, and we form with the help of bacteria. This is all about tools and technology of biotechnology process. Okay, and now we come to the application of biotechnology. So, next class we will discuss about the application, how to apply. So, okay, thank you.